Hi, I'm Serena. I'm from Imperial College. Yeah, so there's been a lot of introduction about collective behavior and C. elegans already today, so I'll keep that introduction brief so I can talk more interesting things. But yeah, so like we have introduced from the beginning of the session, motility-based collective behavior have been studied in different kinds of systems, from microscopic scale bacteria and cellular systems to macroscopic scale fish, bird flocks, and so on. Sorry, can you see my screen still okay so far?
It's not advancing.
Yeah.
If you thought it was advanced, yep, now it's advanced.
Okay, there we go. Okay, so maybe I will just keep this one then.
Yeah, that's fine.
Right, so that was the slide. And then, so C. elegans actually occupies a really interesting position on this scale question, and that they are mesoscopic, and that is one scale that's not looked at very much in terms of collective systems. So they're intermediate both in terms of physical size, because they're just 1 millimeter long, but also in terms of their behavior complexity as well, because unlike fish and birds, they can't see and they don't make sounds, so they're not really relying on long range interactions based on visual and acoustic cues. But compared to bacteria, for example, they do have a compact nervous system, so they are capable of more complex behaviors, in addition to simple molecular diffusion and direct contact that seems to govern a lot of the microscopic scale collective behavior. So the question is really, what sort of interactions matter for C. elegans at this mesoscale? So I guess I can't build this in one by one, but what does collective behavior look like in C. elegans? So Antonio showed you just now, worms crawl around, so single worms can crawl. And tens of worms can actually form aggregates. So they can aggregate into these tight clusters that move around on food. So here they're just crawling on a petri dish with lots of food. And when you have thousands of worms, you can actually get them to swarm. So here, I'm just hearing something. Okay, so thousands of worms can actually form these swarm, and that's really visually striking, as you probably agree. So the worms here form this traveling wave, and it's sweeping over the entire surface that's covered in bacteria. And the swarming behavior actually, in addition to C. elegans, is found in many different nematode species spanning the entire phylum. So it's a relevant, ecologically relevant behavior. So my title slide gave it away in that aggregation and swarming behavior is actually linked. So for the majority of this talk, I will be focusing on understanding the aggregation behavior. So we have a system where we have two contrasting aggregation phenotypes. So the video I showed you just now is what the wild strains do. So if you go isolate worms from the wild and grow them in the petri, that's what it looked like. However, you have thousands, if not tens of thousands of C. elegans researchers are mostly staring down a microscope looking at this lab strain that isn't very social. So this is the solitary strain. We know there's a genetic basis to the difference between these two, because the lab strains are weird. But the question for me at this point is asking, how is aggregation of C. elegans a bona fide collective behavior? And if so, what sort of interaction rules govern the difference between these two strains and their really different contrasting population level phenotypes? So to do that, I use fluorescence markers to just mark the head of the worm, the pharynx, so that we can look inside the clusters, because otherwise they're very dense and it's impossible to track individual worms. In addition, I have a subset of the worms marked with these body wall muscle markers, so that I can track these for a longer period of time as they go in and out. I can also look at body posture. So what you get are videos like this. So up on the upper left-hand corner, you can see a cluster forming, and there are red worms going in and out of that cluster. So worm is now in the cluster, but the red worm will leave the cluster and then rejoins. So what you can see is that the body posture actually looks quite natural inside the cluster, and the cluster itself is pretty disordered. So there's not really a strong alignment of the worms inside the cluster. And of course, we'll quantify all of these. So with raw imaging data like that, we can track multiple worms simultaneously using TRPC Tracker, a software that Abelino Javier in the lab wrote. And we can get centroid tracking data as well as skeleton of the objects, as well as the head to tail orientation of the worms, because they're capable of moving forward and backwards locomotion. Because there's been lots of talks about pheromones in other systems, I thought I'd quickly throw in this slide to say that we did check long-range communication via pheromones do not seem to be important for C. elegans aggregation. So on the top, this is the wild and the lab strain, and these are these two strains. The second and the fourth panel are the two strains with pheromone taken now using a genetic mutation. And qualitatively, they look pretty similar to the original strain with intact pheromone. And you can also quantify them by assessing various things. And yeah, it doesn't seem so. So we focus on local interactions. Yeah, this is meant to build in one by one, but I can walk you through this slowly, so just bear with me. So one local behavior that we can empirically quantify to be different between the two strains is their speed. So on the left-hand side, look at this graph, look at the magenta line, where that corresponds to low local density, so i.e. not very many worms around. This is the distribution of individual worm speed. And you see it's peaking at about 250 microns per second. So the worms are moving at this fast rate. And as the local density is increasing, so moving into this cyan region, so you can see that the speed profile has shifted over to the left, where most of the individuals are now moving at a low speed at about 50 microns per second. And this shift of speed profile is gradual, depending on the local density. And this density-dependent shift in speed also happens in the lab strain that doesn't aggregate so strongly as well, but you can see it's less pronounced. So intuitively, the speed switch promotes the initiation of aggregation, because imagine you're a worm with no worm around, you're moving around really fast, and that can maximize the encounter of other animals. And as you encounter more and more neighbors, you slow down, and that's how you can promote the initiation of aggregation. And then a second behavior that we measure to be quantitatively different between the two strains is reversal behavior. So here with yellow outlining the outline of a cluster, and the purple trajectory is following a worm that's leaving the cluster but reverses back, and it does that again, and now it's doing it again. So this worm left the cluster 3 times, and each time it comes back into the cluster by initiating a reversal, which is a backwards movement. So this kind of reversal, on the right-hand side, you can see the graph. It increases as a function of a local density for both strains. However, the aggregating wild strain reverses more frequently than the lab strain at all densities. And intuitively, these reversals help the aggregates stay together by preventing the worms from leaving the group. So the question is, okay, so now we identify 2 local interaction rules. Are they enough to explain what we observe experimentally? So to answer that, we developed an agent-based model this is a phenomenological model that's purely based on locomotion of the worm. So we have chain-like agent with many nodes connected by springs. We have a front and the back. So the rest of the body nodes are following the head, but each node is undergoing a random walk. So we did some simulations, and by combining just 2 free parameters, which are reversals and speed switch, you can actually capture a diverse range of aggregation phenotypes already. So you can have something that's quite solitary, so like this orange panel that's similar to the lab strain, and you can have something that's more aggregating, so like the blue panel here that's more similar to the wild strain. However, what you can probably see already is that by calculating the summary statistics that describe overall spatial distribution of the objects in space, 2-dimensional space, what you can see is that actually this 2-parameter model doesn't capture the lab strain very well. So there's a gap between the experimental data in dashed line and the simulated data in solid line for the lab strain in blue. Sorry, for the wild strain in blue. For both the pair correlation function and the distribution of hierarchical clustering branch lengths. So we actually had to add in a 3rd parameter that allow us to simulate the wild strains better. So I'll play each one of these movies one by one, but adding attraction as the 3rd parameter allow us to get much tighter clusters that are much more similar to what we observe experimentally.
2-minute warning.
Yep. Yeah, and then the lab string, we were already fine with 2 parameters, but yeah, adding a 3rd parameter was also okay. So then we can recalculate those 2 statistics, seeing that now we have much better quantitative agreement between the 2, between simulated versus real data. So the conclusion here is that at the mesoscale, for C. elegans aggregation, you can actually get that just from local interactions. And these are the 3 rules. So I just wanted to quickly say that this attraction towards neighbors that we added in at the end, that's actually implemented at the range of 1 worm length. So it's still fairly local. It's not like some other systems where this is much more, many body lengths away where the neighbors are. And just with a minute at the end, I wanted to come back to the swarming story. So initially I showed you this video of a swarm, and that's moving from areas with no food to areas with food, which is at the bottom. Okay, so on the right-hand side, I'm about to play this movie, but I'm basically labeling the bacterial food with the bioluminescence marker to show that actually as the worm swarm is coming, there's actually no food left as the swarm front passes, like complete depletion of food. So we tried this with our model. We now start with many worms in a line. This is called periodic boundary conditions. White is food, black is no food. So now each worm has a particular feeding rate, and what you can see is the emergence of the swarm just by adding in this food depletion. So yeah, aggregation plus food depletion gives you emergent swarming in our model, and we're able to say that aggregation and swarming are related behaviors. So we're now developing an easier model, a simpler model, so we can run many more agents at the same time.